Here we're going to start to look at the muscles and the muscular system. So muscles are a unique type of cell for which there are three types, and we're going to mostly in this lecture series focus on the skeletal muscles. But there are also the smooth muscles and the cardiac muscles, which are not consciously controlled and have some unique properties. But regardless of which type of muscle we're talking about, they're all three going to share these main four characteristics. And that is, one, they're all contractile. These are the rare type of cell that can actually shorten or change its length. And we're going to look at how these guys do this, but they shorten forcefully. So they're going to use ATP to do this process of shortening. And then they're going to be able to lengthen back passively. So they're able to change their length from short to long. Lengthening passively kind of goes along with this next one, which is the ability to stretch. So these cells, not only can they contract, but they can also actually stretch back out even longer than they were before. And usually this stretching is either by an opposing force, like an opposing muscle, an antagonistic muscle that's pulling it to be longer, or through gravity. So you can't see my arm, but if I have my arm bent and flexed, and then I allow gravity, if I kind of tilt my body so gravity pulls it, gravity would stretch it back out. So this ability to shorten and to stretch are both unique to muscle cells. And so we're going to have to look and see how they do this. The third property is that they're all excitable. And this is kind of similar to those that we saw with the nervous system. And when we go through the excitability of these muscle cells, it is going to look a lot like what we saw for the neurons. But this is a unique property that muscle cells all have, this ability to respond to stimuli. And then the fourth one here is that they're actually elastic, or they have this ability to recoil. And that kind of goes along with what I said up here with their ability to shorten. But here we're actually saying that they have this recoil, they're going to naturally kind of snap back to their original length after they are stretched. So these are the four major properties that all muscles share, regardless of if we're talking about smooth, cardiac, or skeletal. Though in this lecture series, we're going to focus initially, at least, on the skeletal muscles and how they do these four properties. Okay, so here we're looking at a skeletal muscle because we're going to mostly be focusing on the skeletal muscle. So you can see here it's attached to bone. So this is a skeletal muscle. And what we're seeing looks like many layers, or kind of like nesting dolls. That's what this picture always kind of strikes me as. So I want to help to orient you with what you're seeing here. First, let's find the muscle cell. Now the muscle cell is actually renamed a muscle fiber because here's the muscle cell. I'm going to kind of outline, there's its membrane, and then it's this long, skinny cell. So it doesn't look much like a cell at all, which is why it's named a muscle fiber. It is a cell, but because it's so long and skinny, we tend to call them muscle fibers. And we're going to kind of follow this F sound for a lot of the things within the muscle fiber, as you'll see. So packed inside the muscle cell, or as we just renamed it, a muscle fiber, are going to be many units called myofibrils. And so here we're seeing these units of myofibrils. Here's a myofibril that's actually kind of been pulled out so we can start to see what's inside these myofibrils. And inside the myofibrils are lots and lots of long, skinny filaments. These are called the myofilaments. And so these myofilaments will be myosin and actin, and we're really going to be following them a lot as we get into the sliding filament model of cellular contraction. So packed inside these myofibrils are lots of myofilaments, which are the actin and myosin. And we're seeing many units of myofibrils inside here. So all of these are just packed full of myosin and actin myofilaments. So that's what fills up the muscle cell, or the muscle fiber. Now, the muscle fiber is surrounded by a layer of connective tissue called the endomysium. And so we're going to see several layers of connective tissue. They're all going to be mesium, 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 and we're going to have endo, peri, and epi. The endomysium is the most internal, and it's surrounding a single muscle fiber. Now, in part, it's separating the individual muscle fibers, but it's also connecting the individual muscle fibers. So the different endomysiums of each of the muscle fibers are going to be connected together by this layer of connective tissue called the endomysium. So they're going to, as one muscle cell contracts, it'll kind of pull on its neighboring muscle fiber as well. So all of these muscle cells or muscle fibers are surrounded by this endomysium, and that endomysium connects them together. The individual muscle fibers are going to be packed together in what's called a muscle fasciculi. I'm going to, here's that word a little bit bigger. Groups of fibers are going to be packed together in a fasciculi. So here in this next layer of the next level of the nesting doll, we're seeing these packets, which are called fasciculi, and they're filled with muscle cells or muscle fibers. So inside here, each of these fasciculi are going to be lots and lots of muscle cells, each wrapped with a layer of endomysium. The packets of muscle fibers, which are called the fasciculi, are themselves going to be surrounded with a layer of connective tissue. This time it's called the perimysium. So here's this word, perimysium. So the perimysium surrounds groups of muscle fibers. So there's lots of muscle fibers inside this fasciculi, and then the whole fasciculi is covered in this layer of connective tissue called the perimysium. Many fasciculi will be packed together in a whole muscle. So inside this whole muscle, we're seeing lots of packets of the fasciculi. And then as these get packaged together into a whole muscle, the whole muscle is going to be surrounded by a layer of epimysium. So here I'm trying to kind of outline this epimysium, which is around all of the fasciculi together. And so the whole muscle is going to be surrounded by epimysium. So to kind of keep these layers straight, it helps me to remember that the endo is the most internal, so it's around a single cell. And then the epi is kind of epically around the whole muscle. And then the perimysium is going to be around these bundles called fasciculi. And a fasciculi is a whole bunch of muscle fibers packaged together. Next, we're going to look more carefully inside a single muscle cell. So in this picture, we're seeing the insides of a single muscle cell, and we've just renamed it a muscle fiber, which is another name for cell. So inside this muscle cell, this is a scanning electron microscopy, which is a kind of the best microscopy we have to visualize things. Inside this muscle cell, we're able to see these are the myofibrils. So each of these are bundles of myofibrils, and inside here will be the individual myofilaments or the actin and myosin. And we're able to kind of see around the outside what's called the sarcoplasm and T-tubules. But let me show you these first in a cleaner image, which this is a model image, which is made of plastic, and so everything looks very clear and we can see what we are seeing very well. So let me orient you to what's inside a muscle cell. So in this picture, we're seeing a muscle cell, which is also known as a muscle fiber. And as we look at this cell, there's a couple observations. One, it doesn't look like any other cell that maybe you've seen. But two, it is still just like any other cell you've ever seen. So we're still going to have a nucleus inside the cell. We're still going to have lots of mitochondria. So these little red guys over here that I'm trying to circle are all the mitochondria. In fact, a muscle cell is going to have a lot of mitochondria because it needs so much energy. And in fact, it may have more than one nucleus as well. But what happens is these organelles, the nucleus and the mitochondria, kind of get shoved to the exterior of the cell because in the middle we have all of these packets of the myofibrils inside here. Okay, let me walk you through these different vocabulary that describe the muscle cell. So it still has a cell membrane. However, the cell membrane is still on the outside, just as you would expect, and it is still made of a phospholipid bilayer, just as you would expect. But it's often called a sarcolemma instead of a cell membrane. So a lot of the cellular organelles have different names that describe them and how they're a little bit unique for a muscle cell. So the cell membrane gets renamed the sarcolemma. And I can keep that straight because sarcolemma has got those two M's right here, and so does membrane have two M's. So that just kind of helps me remember, oh yeah, a sarcolemma is just a new name for the cell membrane. And we kind of follow this sarco prefix into the next word. The cytoplasm is still all of the space inside the cell. However, we are going to rename it a sarcoplasm when we're talking about that of the muscle cells. The endoplasmic reticulum, if you remember, for a normal cell, here's the cell membrane, here's the nucleus, and then we would talk about the ER, or the endoplasmic reticulum, being this membrane system that kind of creates these hallways just outside the nucleus. Well, for the muscle cell, it's going to be renamed the sarcoplasmic reticulum. You can kind of see what they did with that name there. And it's still going to be these hallways. We're going to, if you look over in this picture, you can see it in like this manila color. So this is the sarcoplasmic reticulum in this yellow color. So it is still this kind of hallway system, or this tubule system. But it's going to have a unique function that we're going to have to follow. A little spoiler is it's going to sequester or capture calcium, and so calcium will be packed into the sarcoplasmic reticulum. But we'll get to that momentarily. Another thing that's highly unique to the muscle cell is T-tubules. T-tubules are inward extensions of the sarcolemma, or the cell membrane. And so if we look carefully in this picture, here's the sarcolemma on the outside, and then here we see the sarcolemma, or the cell membrane, invaginate. And so it's kind of denoted in this blue color once you get a little bit further. This is the T-tubules. And so what the T-tubules are doing is they're bringing any kind of fluid that's outside of the cell can now be filling these inner compartments of the cell. So this is going to be important when we talk about the action potential, because we're going to talk about muscle cells having an action potential, and just like with neurons, that action potential is going to spread down the cell membrane. However, what's unique for these muscle cells is when the action potential spreads down the cell membrane, it will also be able to spread into these T-tubules. And that's going to be really important. Another thing that you can see here is that these T-tubules are nestled right next to the sarcoplasmic reticulum. So this proximity between the T-tubules and the sarcoplasmic reticulum is going to be highly important. Just to give you a spoiler on this, remember I said the sarcoplasmic reticulum sequesters the calcium? Well, that calcium gets trapped in the sarcoplasmic reticulum, and it's going to be this action potential spreading down the T-tubule membrane that's going to cause a voltage change, that's going to cause voltage-gated channels in the sarcoplasmic reticulum to open and spill calcium. We'll get to all that later. For right now, you just need to understand what the T-tubules are, and that they're going to be nestled really close to the sarcoplasmic reticulum. Okay, and then if I kind of jump back to this picture that we saw a minute ago, now that you've seen the sarcoplasmic reticulum and the T-tubules, let me point out where you're seeing them in this kind of crazy picture over here. The sarcoplasmic reticulum is this kind of white, wispy stuff. And then the T-tubules, you can a little bit see them kind of nestling across the top of these myofibrils, these lines on the myofibrils. But now you can see kind of also why this other picture is much cleaner. We can see things a lot clearer. To keep going down the list, the nucleus, I've already pointed out, is over here kind of pressed against the membrane, pressed against the sarcolemma. And there actually could be many, many nuclei along the length of a muscle fiber, because muscle fibers can be really long. If you think about a muscle in your body that stretches along the length of like your femur, you have to have muscle cells that are as long as the muscle. So they can be the length of one of the long bones of your body in order to be long enough to be that muscle. So because the cells are so long, it makes sense that there might need to be more than one nucleus along the length. The nucleus is important because remember it has all that genetic material that allows the cell to make proteins. And obviously the cell needs lots of proteins because all of these myofibrils are made up of the myofilaments, which are proteins. Myosin and actin are going to be proteins. So the cell needs to be able to make lots of things in order to work. So there needs to probably be more than one nucleus. Also, same for mitochondria, because this is requires so much energy for muscular contraction. There's going to be a lot of mitochondria as well. But largely, the sarcoplasm, meaning that cytoplasmic space or the space inside the cell, is going to be filled with myofibrils. So let's turn our attention to those and see what these actually look like. So here we're looking at a myofibril, and so here's this bundle. This is the muscle cell, okay? So this is the sarcolemma, and then inside the cell are all of these myofibrils. And if we kind of pull out one of these myofibrils so we can see what's inside, that's what we've done here. And now we're looking at just one myofibril or bundle. And as I said, it's filled with these filaments, myosin filaments and actin filaments. So these are called thick and thin filaments. And they're going to be arranged in a repeating pattern called a sarcomere. And the sarcomere is going to, because of the repeating patterns of myosin and actin, will have these stripy patterns. So in this picture up here, you're kind of seeing that this muscle has a stripy pattern. And I'm going to try to highlight it. See, there's a dark band here, and then there's a lighter band, and then there's a dark band, and then a lighter band, and then a dark band. These are called striations or stripy patterns. And you see it in any meat. So when you go to the grocery store and you look at steak, you can see the striations in the meat. And what these striations are caused by, they're in the myofibril. So here we're seeing the stripy pattern. Here it's dark, and then it's light, and then it's dark. These are the striations, and they're caused by the overlap of the myosin and actin. So if we kind of look in this picture, here we're seeing myosin right here. Yeah, I'm just making sure I was right. This is myosin. And then in this lighter color, we're seeing actin. So in this area, we have a lot of overlap between the myosin and the actin. So here it's going to be very dark. And over here it's going to be very dark because there's a lot of overlap between myosin and actin. But in another position, such as right here, we're only seeing the actin. So we're only seeing the actin coming down this area. So this is going to appear to be a lighter color. So where you have a lot of overlap gives you the dark bands, and where you have a little overlap is going to give you that light band. And this is the striations, the stripy patterns that you see within a muscle or within meat. It's actually because of the overlap of the actin and myosin within the myofibrils that fill the sarcoplasm or the cytoplasm of the muscle cell. Perhaps you can see those striations a little bit better in this picture. So once again, in this great big image, we're seeing a whole muscle cell. And within that whole muscle cell, here's that sarcolemma on the outside. Inside this whole muscle cell are lots of these packets of myofibrils. And inside each of these myofibrils are going to be lots of actin and myosin. So that's all these kind of stripes that you're seeing inside here. And this gives us that striation. These dark bands, all this being dark and this being dark, and the light bands being the lighter color. And this is because of the repeating units called sarcomeres. Here we've kind of sketched out what a sarcomere actually looks like. And what I want to do is kind of teach you real quick how to draw a sarcomere. What a sarcomere is it's going to be these collections of thick and thin filaments. The thick filaments being the myosin and the thin filaments being the actin. And how they're arranged is you have Z discs, or it's called a Z line here. And a Z line is this protein that makes this Z disc. It's an anchoring protein. And so if I were to sketch this out, I would first kind of sketch out my Z discs. So, and I like to draw those as kind of a zigzag because that helps me remember that it's a Z disc. And attached to the Z disc will be the thin filaments called the actin. So attached to the Z disc, I can draw in, these are my actin filaments. And I remember that the actin goes with the Z discs because when you have Z and A together, it spells za, which is pizza, and I love pizza. So that's my Z discs with the actin attached to it. Then to finish drawing my sarcomere, I need to draw my myosin. So I'm going to pick a different color. Myosin is going to be attached to another anchoring protein called the M-line proteins. And so here's my M-line, and M goes with myosin. So attached to the M line are going to be my myosin filaments. So here's those myosin filaments attached to this. And this kind of gives you a picture of the sarcomere. There's a lot more details that we're going to fill in on this in the next video as we look at how these contract. But we can see some of these banding patterns, and these bands have names. And so over here you can kind of see, okay, there's something called the I band and the A band, and then here's another I band, and then there's something called an H zone. And these different areas are describing the degree of overlap. So the I band is defined as the area where you have the thin filaments or the actin filaments alone. So this over here in my picture, this is the I band. It's going to be a lighter color because it's only the actin, and the actin is the thin filaments. So it just looks like a really light color because they're thin and they're kind of sparse. So that gives me my I band. The A band represents the area where you have myosin alone. The A band represents the length of the myosin. And so we have an A band throughout this length of the myosin. So that's going to be our A band. And then finally, the H zone is this area where you would have myosin alone. And what's going to happen when the muscle contracts is our H zone is going to go away. Meaning when you look at this picture up here that I drew, currently there is an H zone. It's this area where you can only see the myosin. Remember, the A band is the full length of the myosin, but the H zone describes the area where we can see just myosin alone. And what's going to happen when the muscle cell contracts is we're going to increase the amount of overlap. We're actually going to pull the Z discs toward the middle. And when we do, I think this picture down here actually does a better job. Because we can follow, here is the Z disc, and here is the Z disc. So this represents the edges of the sarcomere from Z disc to Z disc. And here's the H zone currently. And what we're seeing happen as we move from this picture to the bottom picture is we're watching the muscle contract. And so as the muscle contracts, what happens is the Z discs move toward the center. And so in this bottom image, the Z discs are much closer together. And what has happened as they get closer together is we lose the H zone. The H zone being an area where you had myosin without any overlap of actin. What's going to happen when the muscle contracts, these Z discs are going to come toward the middle, and we're going to have the actin completely overlapping with the myosin. So we're going to lose what's called the H zone. We're going to lose the area of myosin, these myosin fibers not being overlapped. And because as we move the Z disc closer, we're going to see a total overlap. And so the H zone goes away as the muscle contracts. I think some of these contracting, watching this happen, you almost need to see it as a video. And so I've linked in several YouTube videos where they're actually showing the Z disc closing in order to show that muscular contraction.